2, 3.
Oh, very good. All right. Well, welcome. Thank you for coming today. We are going to start a new class here. It's called Anatomy, or actually I'm calling it Anatomy and Physiology. The book we're using is The Human Body, Fearfully and Wonderfully Made, and by Dr. J. Wiley. And I am so glad that you guys are here, and I am really excited about being able to present this information. I really enjoy studying anatomy, and I've had several different types of classes dealing with that. So this has been, again, a good review for me to go through the information again. And what I want to do is go ahead and pray first, and then we'll get started into Module 1. Okay? Would you pray with me, please? Heavenly Father, we are so thankful that you provided this opportunity for us to gather together in your presence so that we can study one of the most fascinating thing that you created, which is the human body. Lord God, we are just so thankful that you're giving us this opportunity to learn more about your marvelous creation. And I pray that it enriches all of our minds and our lives as we study how you have created so many things to work together, and also realizing that we're just scratching the surface. We have no concept of how wonderful it really is and how intricately everything is together, but you've given us minds to learn and study, and we do have a small base of knowledge that we can rely on. So Lord, I pray that everyone is excited about this, what they're going to learn. And I pray, Lord, that you open our ears, open our eyes, so that we can take in what you have for us. We ask for your mercies on us. We ask for your blessings. In Jesus' name, amen. Okay, again, welcome. Has anybody ever studied any anatomy before? You probably had some in biology, which was a prerequisite to this course. And if you had that, as you start going through this, you can see how it really helps. It's just a building block. We started with biology as kind of an overview of a lot of things in the world, and anatomy. We're looking at specifically human anatomy, although you could study the anatomy of worms or butterflies, anything in particular, because each are fascinating in its own right, but we are focusing on human anatomy in this class. And along with that goes physiology. So what's the difference between anatomy and physiology? Who can kind of give me a little idea of that?
Well, anatomy is parts of the body, and physiology is what they do.
What they do are, why does it work? Okay, we've got a heart, boom, 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 it's beating. But that's the anatomy, we got a heart, right? So you can study the parts of that, but then the physiology of that would be, how does it actually work? And what does it do? So you really get into the meat of what's going on with there. So we also have gross anatomy and microscopic anatomy. And what's the difference between those? It's pretty obvious, it's not really too hard to figure out. Go ahead.
Gross anatomy is parts of the body that you can see without a microscope, and then microscopic anatomy is what you have to use a microscope for, like cells.
Right, get down to stuff that we can't see with our eyes. So gross is anything we can see visually and actually maybe feel to some extent. And then microscopic, we need a microscope to see as we break it down into smaller tissues and things like that. We just can't define with the naked eye. So what are we going to study here? Gross anatomy or microscopic anatomy in this class? Gross. And we're going to both. We're going to do both, right? Because we're going to look at some cell structure and what's inside those cells. So we're actually doing both. But a lot of it is definitely the gross anatomy part of it. Back when I was in school, my first class was anatomy. I had a class in that, and then it progressed on for there. I had a class in kinesiology where we went over a little bit more, and that's looking at the study of movement and with the body and how all that goes in. So we study the muscles and nerves and where they're connected on the bones and those type of things. And then I had some other biomechanic classes, and then I finished up with a gross anatomy class and looking at predominantly not the cell structure, but really in detail of the muscles, nerves, organs, and how they affect. So each one of those builds, and it's so fascinating. I'm just so excited about this topic. Okay. We break the body down into different systems. There's a lot of different systems within the body that we'll be looking at. And there are 11 different organ systems. And that sounds like a lot when you look at the body and say, ooh, 11, how are you going to get 11 out of there? So we had we're going to list them now. and that's the integumentary system, which is what? That's a big word there, integumentary. Anybody know what that is? That is our skin and our hair. That's a system in itself. And what's a big function of skin? What's it do for us? Protects us from so many stuff. All these little bugs and germs that are floating around in this room that we can't see, they're wanting to get inside and just destroy the body. But it doesn't, because we have a skin system that's intact, and it's going to help protect. That's the first line of defense there. We're looking at the skeletal system. Obviously, that's the bones that so give us support. Bones and cartilage. We have a muscular system, and the muscles are going to allow us to do what? Move. Mobility. Nervous system. That's our brain, our spinal cord, and all the nerves that go out through the whole body. So that is our nervous system, and that does what? Kind of just
Feel stuff.
you can feel things. Pain. Regulates things that go on in the body, because it senses and it might have to send impulses down and cause things to happen. We're going to get into all these in a little more detail. I'm getting kind of a real big overview right now. The endocrine system, what is that? Glands, right. Little hormones and things that get shot out through the body that's going to regulate certain levels of things in our body that need, and if they get out of whack and out of kilter, then the body's not functioning appropriately. Then we have the circulatory system. That's composed of the heart, arteries, veins. We have a lymphatic system. The lymph nodes, you guys have heard of those, I'm sure. And the spleen, little lymphatic vessels in the body, and that's to help kind of drain things out that don't need to be there, and that's to get rid of them out of the body. Digestive system. That begins at our mouth and down through our intestines and our stomach. Well, stomach and intestines, I'll get them out of order there. The liver and those type of things. The respiratory system. How we get oxygen in and out of the body and other gases and things. And then we had the urinary system, kidneys and bladder, and then reproductive system. Okay? So, those are the 11 systems that we're going to study as we go through this course in anatomy. And now let's look at a few things. What is an organ? Besides what you're at church on Sundays, and people play that. What's an organ? Organ in the body. What's an organ?
An organ is a group of tissues specialized for a particular function.
Wow, that's not a good definition, is it? Very good. All right, give me an example of an organ.
Like your
heart. Good. Very good. All right. How about, so it's made up of specialized tissues. So what's a tissue?
There's things that are made up of cells.
Cells. Good. They're specialized cells to come in and make up a tissue. And then cells are made up of what? What's inside of a cell?
Organelles.
Organelles.
And they're made up of molecules.
Right. All right. You've got it down there. All right. Very good. All right. There's 4 basic types of tissues in the body. Now we have a lot of different, because we know we got kidneys. So is there kidney tissue? We've got a heart. Is there heart tissue? Is there? Yeah, in a sense, we do call them generally tissue, because when we talk about the kidneys, we'll talk about the tissue of the kidney, all right? But there's really 4 basic types, because there's bone, there's skin, there's this and that. So there's not like, 11 or so different types of tissues. There's only 4. We have nerve tissue, and our brain obviously is made of that, our nerves, our spinal cord, and our eye. Those are all nerve tissues, all right? And we have muscular tissue. Obviously, muscles are made up of that. We have connective tissue. Give me some examples of what connective tissue.
Like tendons.
Tendons?
Or ligaments.
Ligaments? Come on, another one. Tendons, ligaments, connective tissue. Cartilage, yes. That's in there too. Very good. There's another big one. Very hard substance in our body. Bones. Thank you very much. Thank you very much. All All right, that's a connective tissue. And then we have epithelial tissue, and that's going to make up most of those other kind of organ tissues that you're going to be thinking about. The glands, the skin, lining of the mouth, all the linings of all the tissue, all the organs are epithelial tissue. And so as we study, look at several different levels in the human body. So we've got a human organism. I guess I'm an example of that. We have organ systems. We went through that, those 11. We have organs in the body, and those organs are made up of tissues. So once we get to that level of tissue, then we're starting to look at the microscopic aspect of anatomy. Okay, those first ones we're looking at gross anatomy. Then we have cells, and then we have organelles, which Tim talked about earlier. And remember that organelles are membrane bound. They're enclosed in a membrane, and those are eukaryotic cells. Remember that from biology? That was like chapter 2 or 3 or something like that.
There was prokaryotic and eukaryotic.
Right. Seems like chapter 2. I can't remember for sure. And that won't be on the test, so you don't have to worry about what chapter that was from. So we have then those tissues, or excuse me, organelles, they have molecules in them, and there's proteins and salts and fats and acids and all kinds of stuff in there. Okay. So as long as everything is going perfectly in the system, everything is working right, that is a state of homeostasis. Remember that word from biology? Homeostasis, everything is working perfectly. So the goal is to keep that in equilibrium. Everything going. The body functions much better when everything is in equilibrium. But does that happen all the time? Now, give me an example of something that could throw a system out of homeostasis.
When you're falling from like a building.
Falling from a building, that would definitely put you out of sorts. Yes, it could be very simple. A cut in the skin, no longer in a perfect state. There, we've got damage. So hopefully,
not getting enough sleep.
Not getting enough sleep. That can throw you off, right? It can throw. You're not
Just like laying down.
sleep deprived. What was that one?
Like laying down and then just like standing up.
Laying down and standing up. It could be if things are out of sorts. Say your blood pressure is not regulating fast enough, you get real dizzy, woozy, and pass out. So you're not really in homeostasis here, are you? You're going to pass out. All right. So the organs will stay in a state of homeostasis until it is stressed. Okay, a stress is put on to the system somewhere, and a stress will disrupt body functions and body levels of things. We talked about one of our systems is the endocrine system, and that system deals heavily in trying to regulate and keep us in homeostasis, keep us in a state of good function. There are different levels of chemicals that need to stay in balance within our body system, and the endocrine system plays a very vital role in releasing hormones that will keep everything kind of in check and keep you where it needs to be. And our nervous system, one of those 11 systems we talk about, is also there to help detect where these levels are by having little sensors throughout the body and in the organs that says, are we where we need to be? And they're going to sense those changes and cause an effect on hormones to be released to help regulate that best it can. And sometimes we really get out of whack and things can't get in line, and you get kind of ill and you have to go to the doctor, and then they run a bunch of tests. They're testing for these levels. Where are they? And when they find out if those levels are in check or not, then they will administer drugs at times to stimulate that process and giving you what you might need. And there's some people that need to continue on with those medications because their body's not able to regulate those levels where they need to be. So medicines sometimes are brought in to help with that. So we need to try our best to stay in balance here, or homeostasis, so that we can function at our optimum, right? The hormones would be called an effector. The word effect means it causes an action. So an effector is I have it written down here, nice another definition, is a structure in the body that can change the value of a variable. Make a change on a value or level that we need to be in. So we need a certain amount of sodium in the body, then there's not there, then the body's, because it's getting wasted and through another area. As that sodium comes in, there's going to be some regulations to try to retain sodium so it's not excreted out of the body. So there's little controls there. So the control center is basically our brain. And it senses changes through those receptors that we talked about. And the brain's going to send a signal to an effector, a source for hormones to be released, and then to cause a positive change in the body. Okay, now we're going to go back and break down and look at the cell a little bit. So we'll go back to what we had in biology and looked at that, but we're going to go down and look at these little organelles that are inside the cell as our review. Okay, first thing, a cell is enveloped in a plasma membrane. And that plasma membrane is there for kind of what purpose? What's the main purposes of that plasma membrane?
To protect the cell.
Protect the cell.
Keep it together.
Holds it together. That's a good thing. So everything just doesn't go floating off. So it's got to contain it so that everything is there together. And then protect it by how?
By not letting things that are not good for it in.
Right. It controls what goes in and out of the cell. So it's going to keep out things it's not supposed to have, and it's going to retain things inside that it needs, and it's going to allow some good things to go out and some good things to come in. So it kind of controls what comes and goes. Very good. Then we have a nucleus inside the cell. And what's the nucleus? What's its big thing? DNA, control center, right. It's going to control everything that's going on. So it senses what's going on, what's needed, and it's going to cause things to be produced that it needs and says what's going on. So you said DNA was inside the nucleus. And does anybody remember what that old DNA stands for? Deoxyribonucleic acid, right? Okay. DNA is so much easier to remember, and that's fine. And okay. I was going to say something. We'll hold that off to later when we actually get into that. Okay. Then we have cytoplasm. What's the cytoplasm?
It's like a jelly substance that's like keeps things floating around.
Right. It's kind of the gel that everything else is floating around in. So it's kind of like the medium that it's in. And you've got all these little organelles floating around inside the cytoplasm, and you've got all kinds of other little chemicals floating around in there too. Okay. Then you have ribosomes. Ribosomes were for what? Proteins. So they make proteins, right? So we have ribosomes that are kind of free floating in the cytoplasm, and we also have them on another structure. What was that other structure called? Ribosomes are also kind of connected to what? I'm sorry? Yeah, endoplasmic reticulum, right? We had that. We have rough and smooth. So the rough endoplasmic reticulum has ribosomes on it, and that's why they call it rough. Very good. And then we have smooth ER, or endoplasmic reticulum, and that has no ribosomes on it. Okay, so the ribosomes are free floating in there, but a lot of them are also on the rough ER. And when I say ER, that's what we're talking about there. Just shorten it down a little bit. Okay, so if a cell has a lot of rough ER in it, what's it going to do a lot? What's going to be one of the functions of that cell?
To make protein.
Make a lot of proteins, right. It's going to be a protein producer. And if it has a lot of smooth ER instead of rough, that tells, now you got to think, what does the smooth ER do?
Makes lipids and carbohydrates.
Right, lipids and carbohydrates. So kind of if you're looking at a cell through a microscope and you see a lot of rough ER, then you kind of know it's a protein producer. If you see some smooth ER mostly in there, then it's going to be for the carbohydrates and lipids. Okay? So that kind of gives you a little clue as you break down and look at that. Okay, we have a Golgi apparatus. And who remember, I always forget what they do for some reason.
They like package things that are being sent out, and they just send it out of the cell.
Right. Very good. So they take chemicals and things, and they do a lot of things. They secrete. They secrete out substances. Okay, we have secretory secreting vesicles. What are they going to do? They're going to be part or tacked in with that Golgi apparatus, right? Because we just kind of said they kind of go hand in hand. So they're going to be, the Golgi apparatus is going to be making the secretion, and these vesicles are actually going to be carrying it where it needs to go. Because it just doesn't make it squirted out there in the cytoplasm, because it's going to just disperse, right? It's going to get kind of wasted. So it puts it in a little vesicle or a sac, and that can float to where it needs to go. And don't ask me how it knows where to go, because I don't know. That's for God to figure out. And I'm sure maybe somebody knows how they figured out some of that, but you know, he's designed it, and it says, I need this, it makes it, it goes there where it needs to be. Okay? So we'll just leave it at that right now. So if you ask me a question like that, I'll answer, I don't know. Okay? We had an example here in the book it gave about insulin. So the Golgi apparatus and the pancreas is going to make insulin, because the body needs insulin for what? Does anybody know?
Processed sugar.
Processed sugar. It gets sugar into the cells, gets it into where it needs to be. So we have insulin that's needed. So if the pancreas is not producing insulin, what do we call that medical condition? Diabetes. Thank you. So we have an observer in the back. I'm trying to tell her not to answer that question. All right, that's good. All right, so anyway, the Golgi apparatus within the pancreas is going to be producing this insulin. It's going to put in a little vesicle, and it's going to be floating there, waiting to be needed. And when it needs it, the body's going to take it out, put it out through the cell, and gets it out into the bloodstream. The bloodstream's going to take it, and so it's getting into the cells that needs it for energy. Okay. Let me see, where am I here? Okay. Oh, we have lysosome. Lysosome. Their main function, lyso. Remember that kind of prefix lyso? Lysis? What does that mean? You remember? From biology, we talked about that one. It means to break down. Break down. Okay. So its main function is to break down lipids, proteins, polysaccharides. Remember, what were polysaccharides?
They were like cells that have like multiple. What was it?
What's a saccharide?
It's a lipid, right?
No, lipids are fats, but saccharides, sugar. Okay. So glucose is a simple sugar. Polysaccharides have a lot of sugar, big complex sugar, and it needs to be broken down. Okay, so it's going to be lysed. Lysosomal action on it is going to break down those polysaccharides to mono and disaccharides so they can be absorbed into the body, because a polysaccharide can't be absorbed and to be used for energy. So it's stored that way. But when it's needed, it's got to be broken down so it can be used into glucose or a simple sugar. All right. So you guys are doing great, by the way. We're going back to last year's biology class. You're doing a great job of this. Okay. Mitochondria. What does that mitochondria do in there? What's its big thing? Energy. The powerhouse, remember that word? Powerhouse of the cell? All right, it produces ATP. Okay, we asked what DNA was. What's ATP? I can't remember, that's why I'm asking a question. Triphosphate, something, adenine triphosphate? I've got it written somewhere later on. All right, anyway. Hold me accountable to that, okay? Hold me accountable. We'll come up to it here in a little bit. Anyway, ATP is produced into there, and energy is stored in that ATP. And when the ATP is broken down into ADP and a phosphate, we take out one of the phosphates, then energy is released. All right, so that's how the body releases energy. And we're going to get into, again, the production of ATP and all that kind of stuff in a few minutes. Okay, we have centrioles. Remember what those were? They had a small section that we actually went over, but they have a kind of a major function when we go into cell division. Okay, and we're going to get to that a little bit later on in the chapter too. All right, so during meiosis and mitosis, the centrioles, remember there was two of them in a cell, and they went to opposite ends of the cell, and then then they pulled the chromosomes apart, right? All right, okay, and then we have cilia. And those are cells with little, we remember the cilia, little hair-like follicles on there. Now we find those in the trachea, airway, and hair. I thought of something that's, here we talk about gross anatomy, so we're talking about something that might be a little gross. And another term, definition for the term gross. Cilia in the lungs, okay, we have that in the trachea, and what's it there for? Does anybody know? What's the cilia in the...
Keep water and stuff out of the lungs.
And stuff, yeah. So it's kind of mucousy in there and everything, and this is where, you hear your parents, you guys are a little younger, they're always clearing their throats and stuff like that. As you get older, a little more gunk gets down in the lungs, and it has to come up. And the cilia is working hard and it gets it up. So when you're younger, you don't hack up a bunch of stuff, but when you get older, you kind of do that a lot. And it always annoys the kids. Oh, there they are again. But anyway, the cilia is there to get all that stuff out of the lungs, and it needs to be gotten out. And it brings it up so that you cough it out, and then you dispose of it. So anyway, that serves a very important purpose. Okay. Let's look at protein synthesis. And then we had our gross part of gross anatomy. All right. Okay, proteins, they're large molecules, and they're formed by joining amino acids together. And the type and number of amino acids there are is going to determine what the protein is and what it does. So proteins are very complicated and complex. They're a very large string of these amino acids together, and they are produced. And who can tell me where they're produced? We already talked about this.
Ribosomes?
Yes, very good, and the ribosomes. And so we're going to look at how that happens. Protein synthesis takes place in two steps. And the first step is called transcription. Do you guys remember what happens in transcription? We're looking at the cell nucleus. We have the DNA in there, and it's in a double helix, and it starts to unravel. All right, so that's our DNA, kind of unravels. And what connects that double helix together? They were
the mRNA.
Okay, you're a little ahead of me. I backed up a little bit, so let me back up again and ask the question. And that double helix, it's connected along that double helix by nucleotides, right? Okay, and there's how many types of nucleotides? 4. Good. Okay, what are they?
I forget.
One starts with an A. Adenine.
Oh, yeah.
We're just going to use A.
Thymine.
And
Cytosine. Yeah.
cytosine
Yes.
and
Guanine.
G. Okay, and now these only connect with each other, right? A and T's connect, T and A connects. C and G connects, G and C connects, right? But G won't connect with A, right? This is our little pattern. So down along that double helix, there's all these little letters, if you will, and those are the amino acids, and they're the nucleotides. I'm sorry. And as they connect down, as they go along this string, and we start connecting much of it together, then we form a protein by all of that. Okay. So now we're going to jump back a little bit to the question there. As that double helix unwinds, something else comes in, and that was
The messenger RNA.
messenger RNA. Okay, so that's different than the DNA. So that's ribonucleic acid comes in, and they're going to line up. Okay, we can leave that up there. I'm using red, and we could have this pattern. I'm just going to make these smaller. This is still in the double helix, okay, and we might have T and an A here. So when this unravels, let's say that is exposed. Got my colors here. All right, so this is a little chain of, let's go here. 1, 2, 3, 4. Okay, I got 6 of them up here. Okay, so we have this exposed now, right? So the double helix is unraveled with this exposed. We have the RNA come in, and it needs to connect to that for the transcription to happen. So it transcripts or copies over. So what's going to connect here as we go down the list? You guys tell me what connects. What's going to connect with the T? A. What's going to connect with the A?
Uracil.
Uracil. Why?
RNA doesn't have guidelines.
Right, exactly. So what's going to connect here? G. You can just say the letters. This is a C. Can you guys see that? I'm sorry.
Yeah.
Okay. G. We got an A here. U. And you got a T here. A. Okay. So this is the DNA up here, and this is RNA. Okay. So this stuff gets transcribed. So it's a code. It says, okay, we got this. This is what I need a protein made of. So I do this little transcription here, and then where does this go? Goes out to the ribosome. Very good, very good. And why is it going over to the ribosome?
To translate and make a protein.
Exactly, it's going to go make a protein. So this guy goes off, and this is called RNA as it comes in, but it also gets another little letter attached to it. Little M. Wait, what's the M for? Messenger RNA. Okay, and it goes over to a ribosome, goes into the ribosome, and it will line up again with a partner. So let's say, let's do this. So as it goes over, this was a, okay, I'm going to put it in line. So we have A, U, right? G, U, A, right? So that goes over to the ribosome, and it goes in like this, and it has to line up to be translated, right? And it's going to be translated into what?
The transfer DNA.
Yeah. And it's going to line up with A and U, okay. And that's going to make the protein. And it will keep producing out until it's got all the code that it needs for a particular type of protein. It's going to come out the other end. So it keeps adding these chains on there, and it spits out. I'm just kind of imagine it coming in one side of the ribosome, and it kind of comes out the other end all matched up. That's kind of my mind. That's the way I get a hold of that in my head as it comes out, and it will string along all the appropriate amino acids together to get, and then it will cut it off and say that's the protein I need, and then that protein goes off to where it needs to go to. You guys did a good job of that. That was a good quick review. I don't know if it's going to ask this on the test, but in between the A and T and the C and the G, the bind is called a peptide bind, peptide bond. Excuse me, peptide bond. I don't know if that's in some of your test questions or not. I just thought I'd throw that in. Okay. Now we are going to move on to. Well, we're going quite a ways here. Okay, we're going to go into mitosis. And this is for cellular reproduction. So when a cell needs to be replaced because it's injured, or you just need it for growth, the cells have the ability to grow, and they go through this process called mitosis. Right? Now we went through that really heavy in biology, so we're going to take a quick look at it here. And remember, there were 4 phases of that. Okay, and the first phase was called, anybody remember? Prophase. Okay, and so what's going on during prophase?
The DNA, copies itself.
Okay, DNA is being copied.
The centrioles are splitting and then spreading out across the cell.
Excuse me here. Okay, so let's put some little X's are for what?
The copy chromosomes.
Okay, and we had the centrioles going out, and they're sending out some
Strings.
They're starting to send out some little strings. What are they called? Microtubules? Okay, so that's prophase. Centrioles duplicate, and they begin to form spindles. So they're duplicated spindles, and they're starting. Actually, they're close together. I kind of spread them out a little bit, and they start moving to the opposite ends of the cell, and the DNA starts forming chromosomes. All right, so they're duplicating themselves. And how many chromosomes are there?
In the human body.
In the human body, yes.
46.
46. Okay, very good. Okay, the next phase is what? Metaphase.
They all line up.
So we have these chromosomes starting to line up in the center, and these microtubules that are coming out and starting to latch hold of those chromosomes. So they don't have to go finding them. They come and line up where they need to be, makes it kind of easy. And then those microtubules are reaching out and trying to grab them. Okay, the next phase is what? I'm sorry? Anaphase. And what happens during anaphase? Chromosomes are pulled apart. And where are they going?
With the centrioles.
Right. Pulling apart. So during the anaphase, they're actually pulled apart at that point. And then we have the telophase, would be the last phase. And what's going on? What's going on there?
The plasma membrane is squeezing it up. What do they call it? Constricting. And gotta make it see.
So we start seeing this little indentation here. All right, and as it's starting to separate, and little chromosomes are being pulled in here. And what happens after that? It's after it's totally split. I'd say that's a solid line there now. It's totally split, and it's separated itself.
Then the chromosomes make the needed organelles.
Right, because remember, as this cell divides, you've got other stuff in there. Right, you got all these organelles we've talked about in there, and they're not going to split up. All those organelles aren't going to split up evenly, are they? And this is going to form a nucleus around the DNA. So I have another nucleus in each one, and all those little other organelles are going to be in there. Now, some organelles may be more heavily in one cell than another, just the way it is when it splits up. So what does it do? Say it's got little ribosomes and Golgi apparatuses and rough and smooth reticulums. So what if it doesn't have everything it needs over here? What's it going to do?
It's going to make them.
It's going to make them. How can it make them?
It's going to put the ribosomes, and the DNA is going to control it.
Right. The nucleus is just going to say, what do I need in here? Okay, I just got formed. What do I need? So it's going to say, I need this and this and this. It makes it because it has everything it needs and a duplicate from the chromosomes and DNA to make whatever it needs for itself. So it's going to just make whatever it needs. Okay? Very good. Pretty neat, huh? God's got a good plan. He's going to divide it up. Doesn't quite get all even. No big deal. We make some more stuff. Real short and sweet. All right. Now let's go ahead and look a little bit more at this whole chapter, as you guys have figured out probably as a review. So we're coming up to the plasma membrane. So that's what covers the whole cell and holds it together. We talked about that's its major job is to hold everything in and also allow things to come and go as the cell needs. And the cell membrane or the plasma membrane, if I use the word cell membrane instead of plasma, it's the same thing. I just will interchange those, whatever comes to my mind when I'm talking. So we have two layers of phospholipids. You kind of remember that?
Like a bigger fat, fatty acids.
Right. So it takes 3 fatty acids to make a fat protein. To make a fat molecule. Thank you. All right. So it takes 3 fatty acids. But with a phospholipid, a silica lipid, it's a fat. One of those is gone and it's replaced by phosphorus, and that's where we get the cell membrane. That's how it's nice. So it's not all fat, but there's fat in it, right? So so we have a phospholipid, and what's the cool thing about that?
Got polar ends and non-polar ends.
Got polar ends and non-polar ends. Because of that phosphorus being attached to the other 2 lipid molecules, we now have something that is polarized. And what's so neat about that is that it can be disrupted and it will correct itself because of polarity. Now, when we had biology, it didn't talk about polarity. It used different words. It used hydrophilic and hydrophobic. Okay, so hydrophilic means it had an attraction to water, and hydrophobic means it had a fear of water, a rejection of water, wanting to go away from it. So we kind of looked at those as being a molecule here, my fingers, and we've got 2 layers of that going on here, and the phospholipid sort of had it so that what's in the middle here was the hydrophobic, because if you look at the cell itself, okay, look at the cell, and the outside of it has a lot of water in it, and the inside of that cell has a lot of water in it, okay? In the cytoplasm, there's a lot of water in there. And the insides of that cell wall didn't like water. It's hydrophobic or non-polar. So if it got disrupted and stuff came in, then if it got flipped up, then it would write itself right back and join together, because it's the part of the cell that's on the inside, it doesn't like the water, so it's going to always repel. So it's always going to come down and try to kind of stay and hold itself together there. So because of the polarity of the molecules, they can right themselves, as we just talked about, and reorient themselves. So there always has that plasma membrane there. But there's also some neat things about that, because it allows some things to go in. Okay, there are proteins. These are large molecules within that cell wall of the plasma membrane. And the very large ones are called channel proteins. And that would allow things to come in and out. So it's kind of like a hole. It's made out of protein, but it's kind of like a hole in the cell wall, so that things that are small enough can go right through to get in to the cell or come out of the cell as needed. They will just diffuse in and out as needed. And that's okay. Now another way, we have this glycoproteins, and they act as a marker, or they recognize things that belong in the cell and things that don't belong in the cell. So they're going to make sure the stuff that doesn't belong in there, they're going to try to keep that out. And the things that do belong, they're going to be allowed to come on in. We have what's called receptor proteins in there too. And they take in messages from other cells. An example that they gave of this in the book is, and which we are going to study later on when we get into muscles and nerves. So when a nerve cell comes in and attaches to a muscle, it actually doesn't totally attach to it. There's a gap there. the nerve impulse comes down through the nerve, and it's got to get into the cell membrane of the receptor, which is the muscle. And in order for this, it's a chemical process. In order for that to happen, there has to be receptor cells that will receive that impulse. So those are on the outsides of the cells. And then we also have cholesterol in the cell. And cholesterol is a lipid. You guys have all heard about cholesterol. We shouldn't have too much cholesterol in our body and all that kind of stuff. And that's true. We shouldn't be have our cholesterol levels too high because it does contribute to heart disease and things like that. But we do need an ample amount of it in there. So that the cholesterol there helps to kind of keep the cell together. The fat, it kind of helps keep it all together and intact. So it's very important to have that. All right, we've mentioned that the plasma membrane, it regulates, what comes and goes in and out of the cell. And this is called selective permeability. So the cell is selective of what's allowed in and what's not. And so we have, we talked a little bit about these channel proteins. So small molecules like water, they will float in and out. And by what process will things kind of come and go easily? I'm kind of jumping.
Diffusion.
Diffusion, right. I wasn't quite sure I presented that well enough so you understood what I was kind of asking. But yeah, diffusion. And what happens with diffusion? Why does something diffuse from one place to another? What's kind of the definition of diffusion, basically, what I'm asking?
When the concentration of something is higher on one side of the plasma membrane than the other side.
To the other side. So which way will it go?
To the side that's lower.
Lower. So an area of higher concentration will go or diffuse into an area of lower concentration. So this is kind of one of those laws that God set up to allow things to flow and get to where it needs to be. So a smaller molecule will go through those protein channels, and it will come and go as it pleases or as is needed. And water was an example of a molecule that's small enough to go through there. Fat molecules, they can enter the membrane mainly by it kind of just dissolves through it, because the fat is predominantly what the plasma membrane is made up of, so it can diffuse in and out. I shouldn't say diffuse, it sort of dissolves in and out. All right, then there's also a charge. Positive and negative charges will allow certain things through. So you have some of those protein channels will allow things to come and go depending on the charge or the polarity of that molecule. So if you have a protein channel that's positively charged, it will allow in a negatively charged molecule, but it will not allow positively charged molecules to come through that particular channel. Now you might have some other channels that are negatively charged that will allow the positive ones to come through. So it's not an all or none, it's just an all or none on that one particular opening, okay? Then we have a mediated transport. That's where you need a carrier protein with a specific shape. This example they gave in here, and I don't know if you got your books open, and it's on page 22, where you have this particular protein has a certain shape, and any molecule that comes to that has that shape, it will fit into it, and then it will allow it to come through the membrane. So that's a mediated transport. It's a helper to get it to come through. So things that do not fit that particular shape will not come through that channel. It may go through another channel that has that type of shape. And why would there be different shapes on the same membrane? Just kind of
molecules.
I'm sorry?
Allow for different shape molecules.
Right, so not so different type of things, and there's a couple things here we're going to look at. So there's specificity. It's specific. That channel has a specific function of allowing a particular shape of that molecule to come in. So it's specific. There's also competition. There's only so many ports to go through for that particular molecule. And that's kind of what I was trying to get at. There's some competition there. So, if one is shaped like a triangle, for example, and is trying to go through one that's, shaped like a rectangle, then it's not going to go through. It's not going to allow it to come through, but only the rectangle molecules are going to be able to shoot down that opening. So there's only so many sites for that for competition reasons. Only so many allowed to get in. If it was all loaded with those things, a whole lot of that shaped molecule would get in. And if that particular type of cell needed that, that's fine, but it's not going to allow a bunch of other things in that it doesn't particularly need. And then there's a saturation level. Only so much is going to needed inside the cell. So those kind of three factors control your mediated transport. Okay, let's look a little bit deeper at this membrane transport. We have active transport and passive transport. So active transport would require what? We talked about passive being like the water, can passively come through. It just floats in and by diffusion, right? So there's an opening there, it can come and go. So diffusion of molecules is a good example of passive transport. So what would be active transport?
You have to use energy.
Energy. So ATP. Energy needs to be used in order to do it. All right? So let's look a little bit more at the passive. So we talked about diffusion as one, and that would be going through those channel proteins. It's open and it's just coming and going as needed. And then the facilitated diffusion, which is the mediated transport that we just talked about a second ago, would be another example of not needing energy to happen. It has a particular shape. It comes in to the particular shape tunnel, and it's allowed to just go on through because of its shape. So then active transport would be in need of ATP. Adenosine triphosphate, that's where I wrote it down. Okay, I was close. I don't think I said it quite right. Adenosine triphosphate. ATP is needed, and it's broken down into ADP with adenosine diphosphate so that active transport can happen. And this is a way of trying to get molecules either in or out of the cell that it either does or doesn't need. So if it needs it, it's going to actively have to bring it in, and if it doesn't need it, it's going to have to actively toss it out because it may not be able to just come and go as needed. And that's to maintain the appropriate balance of homeostasis within that cell. So we looked at an example here of sodium. Sodium is a positive charge ion, and it's in higher concentration outside the cell. And it wants to diffuse inside the cell of lower concentration. So it will diffuse in and out. And that's fine to a point. But if too much sodium gets into the cell, the concentration level still might be higher outside than inside, but the cell doesn't need that much. And if it has too much, then the sodium is going to destroy the cell. And therefore, the cell can't have that. So it's got to get some sodium out. Because sodium is going to diffuse, and so it's got to get some of it out. But it's to go against the law of diffusion, it's got to actively push out that sodium. And it's going to do that by using ATP as an energy source to try to move that sodium out. And then the book also gave an example of potassium. It's in higher concentration within the cell than without. So it wants to diffuse out of the cell, but the cell needs it. So it's got to actively transport potassium into itself by using ATP. Okay, another type of active transport is called endocytosis. Does anybody remember what that was?
So bringing larger molecules into the cell.
Right. Endo, you're trying to get it in. Even though it's not IN, it's ENDO, endocytosis. And there's two ways that's going to happen. And those two ways are called pinocytosis, where a protein is dissolved outside the cell into a fluid, and then the liquid kind of lands on the surface of the cell membrane or the plasma membrane, and then it kind of indents itself, and then the cell membrane will wrap around it and pinch it off. So you kind of have a little vesicle of that protein because the protein's kind of large. Remember, it's got all those little nucleotides stacked together, and it's kind of long and big, and it just can't get inside there. So God's designed a way to sort of just dissolve that protein into a kind of a liquid, and it lands on there, and then it engulfs it, and then it uses the protein as it needs it inside the cell. And then we have phagocytosis. We talked about that some back in biology last year, where a cell actually engulfs. So it comes up to, it's almost like a little amoeba. Remember, I think that was the example that we used, was amoebas, where it kind of floats up there, and it will see the little molecule that it needs, and it will go around it, develop a cell wall on the other side of it, and it brings it in that way. So it kind of eats it, if you will. An example they give here is white blood cells. So the white blood cells go out there, and they're phagocytic cells, and they will go out and eat up all the pathogens that are in the body. They're trying to kill that stuff that doesn't belong in there and destroy it. So those are two ways of the endocytosis, trying to get things into the cell. And then there's one more way of active transport, and that's exocytosis, and that's where we transport materials from inside exo. It's trying to excrete it, if you will, trying to get it outside of the body, and that's where we have our Golgi apparatus. Remember, we just talked about that not too long ago, and which chemicals are secreted, and they go into vesicles, and it goes out to the outer, and then it's dispersed out into the blood system. So, kind of a wrap up here. We have the cell membrane is, if it's disrupted, or excuse me, it will be disrupted during the endocytosis and exocytosis. So bringing something in that's large or taking something out that's kind of large, it's going to disrupt that cell membrane because it's going to just open it up, right, and cause it to happen. So that's the reason, the big reason for the design of that plasma membrane to be polar. Hydrophilic or hydrophilic, so that it has an attraction or does not have an attraction of water. So once it gets disrupted, it can realign itself and close back off after that process has happened. Okay, that's the lecture for Module 1, and we have finished that. Now we're open for